Hello students, in the last video we have completed the process of polymerase chain reaction and its application. I hope you understood that. After that we will see the next topic, that is identification of transfer assembly. So before coming to the identification of transfer assembly, why we have to go for the transfer assembly or why we need to identify all those things we are going to see now. So first of all we will see the step by step so that you can understand actually what is the aim. So first of all, first step is introduction of rDNA into the host. So first of all, what we have done, just by adding the some enzymes, breaking lysing enzymes, breaking plasma membrane or the cell membrane, we have collected the genetic material. Then after that, with the help of the restriction endonuclease, we have cut the desired gene fragment. And after that, to find out the desired gene fragment from the rest of the DNA or rest of the genes, what we have done, the gel electrophoresis. After that, first of all, by this electrophoresis process, we have collected the gene fragment, desired gene fragment. After getting the desired gene fragments, we actually allow it to undergo the polymerization reaction so that from one desired gene fragment, we can make the multiple copies. And after making the multiple copies, we insert that desired gene fragment into the host. And whenever we have how we will allow them to enter inside the host with the help of the vector. So first of all, we will collect the vector. We will cut with the help of the same restriction endonuclease. We have cut the desired gene fragment. Then the desired gene fragments will be introduced into the vector. Then make it as a rDNA, and that rDNA will be inserted inside the host body. Suppose this is the E. coli. So how we will introduce the desired gene fragment into the host body? We can have the first step, that is heat and thaw process. Heat and thaw process means we can first of all allow the recombinant DNA into the medium of the E. coli, suppose the host E. coli. Then after that we will increase the temperature for a short period of time, and whenever we increase the temperature, then in the E. coli's plasma membrane, first of all some pore like thing that will be developed, and once the pore is developed, the rDNA will be inserted inside the host body, and at the same time we will add the ice cubes into that suppose specific beaker where we have allowed that reaction to happen. Then what happened, due to the cold temperature, due to suddenly decrease the temperature, the host will shrink and it will take the rDNA into the host body, and that's how we can make the host a transformant host. So this is one process, heat and thaw process. Then the next will be calcium chloride, some type of divalent ion. We can allow the host to increase the pore size, then allow the rDNA to enter inside the host body. Then after that, once again we can make the host a transformant host. Next one, we have the gene gun method. Gene gun method means the method by which the metals, the non-reactive metals like gold and tungsten, that will be layered with some rDNA, and it will be directly injected with a high velocity microparticles, that is gold or tungsten. That is the gene gun method. Then after the gene gun method or ballistic method, then after that we have another method that is known as the micro injection method. In this micro injection method, what happens, we can directly take the rDNA into a micro injection and directly inject the rDNA inside the host body. So this is one process. Next one, high voltage electroporation is a process, isn't that? So this is one electroporation process. So in the electroporation process, what we can do, that the rDNA that which the host will be subjected to high voltage for a very short period of time, within suppose seconds. Then what happen in the host plasma membrane, suddenly some pores will be created, the rDNA will be injected inside the host body, and then after some time, once again the cell membrane will shrink, and this way also we can able to insert our desired gene fragment with the vector that rDNA inside the host body and make the host as a transformant host. These are the steps we will do so that our gene fragment can enter inside the host body. But how can we be sure that that host or that E. coli that is taking the rDNA or not? It is not possible that by any type of strategies we can able to find out the transformant from the non-transformants. So this is one very important steps we need to follow, that we have to check, we have to identify that the host is taking our rDNA or not, right? So that is the step that we are going to discuss now. That first of all, how can we be allowed, how can we be understand that yes, this actually host has taken our rDNA, or this is not taking our rDNA. Otherwise, suppose we don't know whether the host is having the rDNA inside it or not, then after that we directly go for the commercial process, and after that whenever we will collect the desired product, we will not able to get it, isn't that? So that's why or the product formation, whatever the product we need to get, actually we will not able to get that product, because that the whole bioreactor commercially we will give the nutrients, maintain temperature, pressure, work, time, manpower, everything will be there, but whenever it comes to the product, the extraction of the product, we will not get the desired amount of product. So that's why first of all we have to make sure that whether that host we allow them for the commercial purpose, this is taking our rDNA or not. So this is the step first. What we can do, we have completely two steps, two procedures by which we can able to get the transformant from the non-transformant. So the first one, this is antibiotic resistant gene. Okay, so one antibiotic resistant gene. What is the antibiotic resistant gene? First of all, just see in this antibiotic resistant gene, what we will see. First of all, suppose normally we have one E. coli. Okay, so this E. coli don't have any type of antibiotic resistance. Okay, so that's why if we have suppose one beaker and that beaker have the antibiotic, in this first beaker, this is the beaker A, this is the beaker B. In this beaker A we have ampicillin. Ampicillin is actually one antibiotic, and the second beaker that is going to have another antibiotic, that is tetracycline, right? Now after that, if we subject that host, okay, that E. coli, this is a normal E. coli which don't have our desired gene fragment. Then after that, if we allow it to enter inside the ampicillin and the tetracycline medium, is it possible that in presence of antibiotic this E. coli, this bacteria will survive? Never. So this bacteria E. coli, this is E. coli, this bacteria will not able to survive in this antibiotic medium beaker A, and this beaker, that is tetracycline containing beaker B, that will not able to survive. So this is the first thing you understood. Now just see, in this antibiotic resistant gene, what we are going to do. First of all, we have to take this E. coli. E. coli will be the host. Now whenever we make the rDNA, right, then what we are going to do, that in that rDNA, suppose this is the rDNA which have our desired gene fragment. So this is our desired gene fragment, right? So this is the rDNA. In this rDNA, there will be one specific gene, Okay, so this is one gene present here, and this gene is antibiotic resistance gene, right? So this is the antibiotic resistance gene now. So due to the presence of antibiotic resistance gene, suppose ampicillin resistance gene or tetracycline resistance gene, this is the transformant host, isn't that? So this is the non-transformant and this is the transformant host. Okay, so what is the meaning of the transformant and the non-transformant host? If it is a non-transformant host, that will not going to have our rDNA, and if it is going to have this rDNA inside the host, that will be our transformant host. Now just see, if that transformant host is allowed to survive in this beaker A and beaker B, then what will happen? See that this transformant, earlier it was the non-transformant, this is the non-transformant, and now in this case this is transformant and this one is the transformant. Okay, so this transformant have the desired gene fragment, and along with that it is also going to have our antibiotic resistance. So because of this presence of the antibiotic resistance, what will happen? The transformant will survive in this antibiotic ampicillin medium because it has this gene, that is antibiotic resistant gene. Suppose this one is the antibiotic resistance gene, that is either ampicillin resistant gene or tetracycline resistant gene or both. Okay, so this will be our transformant which is going to survive. It is going to survive in this medium. Suppose that antibiotic resistant gene in this transformant is a ampicillin resistant gene. So in ampicillin medium, due to the presence of ampicillin resistant gene, they are going to survive. Okay, and next one, this suppose antibiotic resistant gene in this transformant, the rDNA consists of suppose tetracycline resistance gene. So that's why in this medium, tetracycline medium, this is going to survive. Understand? So what is that? This way it is a clear distinction that we can able to find out the non-transformant from the transformants. Okay. Understand? That means we make the first of all rDNA enter inside the host, make the host transformant, allow it to survive in the ampicillin. If it survives, that means it is a transformant. If it is not surviving, that it is not a transformant, right? So this will be the first step, that is the antibiotic resistance gene. Now there is one phenomenon that is called as the insertional inactivation. Okay, so what is the insertional inactivation means? So there is one rDNA. So this is the rDNA, right? We have rDNA means here we have our desired gene fragment. Okay, so if the desired gene fragment is allowed to be subjected or allow them to attach with the vector in the BAMH site. Okay, so this is the BAMH1 site where our gene fragment, desired gene fragment is attached. So that's why what happened in this, suppose tetracycline resistant gene was there. So in this tetracycline resistant gene is getting inactivated. So tetracycline will not able to work if we allow the gene fragment to attach to BMH1 site. Then the tetracycline resistant gene that is present, okay, as a selectable marker, that is not going to be survived. Okay, so that's why even if we allowed our gene fragment to attach to the BMH site, we are not able to make it survive in the tetracycline medium. In the tetracycline medium, this gene fragment inside the host body, suppose this is the E. coli, that is not going to survive in the tetracycline medium. This is going to survive only the ampicillin medium. So this will be also one indicator by which we can able to find it out that yes, if that host is not able to survive in the tetracycline, but yes, it is surviving in the ampicillin, that means it is a complete successful complete successful technology, what we have done is correct till now. Right, that means this gene fragment which is attached to the vector in the BAMH1 site, the tetracycline resistant gene that get inactivated. So this phenomenon is known as insertional inactivation. So what happened, that host, this is a transformant, but this will not able to survive in the tetracycline medium. This is an antibiotic, so they don't have the tetracycline resistance, so in the tetracycline they are not able to survive. But then also this vector have two selectable marker. One is the tetracycline resistance gene, another one is the ampicillin resistant gene. So due to the presence of, suppose this is the ampicillin resistant gene, they can survive in the ampicillin resistant gene. So this is a good selectable marker, isn't that so? This is the selectable marker. First of all we have discussed is the antibiotic resistant gene. I hope you understand, make it clear so that you can understand this. You just watch this video 2-3 times, because this one is the conceptual thing you have to understand, then only any questions if it comes you can able to do it by yourself. Now the next one, this is the antibiotic resistant gene. So we have completed the first selectable marker. Second selectable marker, this one is the enzyme producing gene. Okay, so this one is the enzyme producing gene. Okay, so this is the enzyme producing gene. What we are going to do is see. First of all, we have to make the rDNA, right? So first of all, we have to take the vector, insert the gene fragment, make a rDNA. So first of all, what we are going to do, see here, this one is the gene fragment where our gene is present. Okay, and this one is suppose this is a normal E. coli, this is non-transformant, this is one E. coli which is a transformant. So this one is the transformant and this one is the non-transformant. Okay, so the transformant has the gene product and the non-transformant don't have the gene product. That our desired gene product is not present. So in this transformant, what happened? See, first of all, we'll start with the non-transformant. In the non-transformant, naturally, obviously the E. coli non-transformant, but then also they are going to have their genetic material or not. Yes. So this lac-operon, if you have not studied till now, lac-operon, whenever you will study, this concept will be more clear for you. So this is one gene fragment in the non-transformant. E. coli have their gene fragment. So in this E. coli, what they have, they are going to have a gene that is known as the LAC-Z gene, and LAC-Z gene have to be present so that the E. coli can survive. They can take the food. Okay, they can survive so that they can eat the glucose, or they can survive. So this LAC-Z gene, they produce one enzyme. This is a gene undergo translation, then after that it produces a protein. That protein is known as beta galactosidase, right? So beta galactosidase it will produce. So this is present in the non-transformant. Then after that we will allow it to work or it to survive in a medium. That medium is called as the X-gal. Okay, so this is one chromosemic substance actually. So this is the X-gal. Okay, oh sorry, this is our non-transformant, right? So this is the non-transformant. The medium is X-gal. X-gal is actually one chromogenic substance, one liquid due to the beta galactosides, because they have the leg Z gene. Non-transformant naturally have their gene product, genetic material where a gene leg Z gene is there. Leg Z gene will produce the beta galactosides, their protein, their enzyme. So this beta galactosides will convert the white color of the egg gel into blue color. Okay, so this medium which have the white color, they will turn into a blue colored liquid completely. So if it convert into a blue color, completely convert white color into blue color, that means what we know, that that gene product, that leg Z gene that is present in the host body, that means we have not injected any gene, our desired gene. Okay, now come to the transformant. In the transformant, what happened, that leg Z gene. Okay, so suppose this is the leg Z gene. In this region, the leg Z gene was there. What we have done, that remove the leg Z from here. So we just remove the leg Z gene. We have injected our gene fragment. So this one is going to be now our gene fragment, our gene product, not the leg Z gene. Right, so leg Z gene we will remove. If the vector have the leg Z gene also, we will remove it. Then after that we will insert our gene fragment in the place of the leg Z gene. That means we just replace the leg Z gene with our desired gene fragment and make the vector as a recombinant DNA. That will be a transformant. So due to the absence of the leg Z gene, is it possible they can produce the beta galactosides? Of course not. So beta galactosides will not gonna produce. So after that, if you insert into the chromosomic substance, suppose X gel, white color of the X gel, will it be converted into blue color? Is it possible they can convert into a blue colored substance, liquid? Never. Why? Because this leg Z gene in the vector is replaced by our desired gene fragment, then make a transformant. And whenever we make it as a transformant host, leg Z gene is not present, the beta galactosides is not going to produce due to the lack of this leg Z gene. Then after that, whenever we will just insert into the medium of X gel, then white color remains white. And if the white color remains white, that means they have our desired gene fragment. Right? So we don't know which host have our desired gene fragment or not. We just insert it into the X gel. If it turns blue, that means the whole procedure is wrong, and if it is remain white, that means our experiment has completed, has done successfully. So these are the two different type of selectable marker. So in the selectable marker, what happens, and yes, this whole method, this is known as the insertional inactivation. Okay, so this is the insertional inactivation method. Okay, that means whenever we insert our desired gene fragment, we just completely inactivate the leg Z gene. Due to the lack of the leg Z gene, the beta galactosides enzyme is not going to be produced due to the absence of it. Then after that, X gel, white color of X gel will remain white. Okay, so this method, this procedure is known as the insertional inactivation. So this way we can able to find out the transformant from the non-transformant. So we have two complete steps. One is the antibiotic resistant gene and another is the enzyme producing gene. We have completed. I hope you understand. This is a conceptual thing. Try to understand it. Read it from NCERT, from the book, from any other references. Try to understand my note also, my video also. You will be able to understand, and once you understand, you will not gonna forget it. So that's the whole step. We have only one video left. That is the bioreactor. That means for the commercial purpose. So that's all about it. I hope you understand. Thank you.